thank you very much. So this is my disclaimer. It essentially says, don't believe anything I say. Go look at our filings and make an investment decision based on that. OK, thanks for having us back. When I was here last year on this stage, I was telling you about why I was excited about the company and what we were doing and where we were going. This is what we looked like. We had a share price of $1.83. Our market cap was about $32 million, and we had less than $5 million in the bank. And so what happens to companies that look like that? I'll tell you, they die. The vast majority of companies that look like that die under the weight of the market. Doesn't matter how smart management thinks they are. Doesn't matter how good technology is. The deck is stacked against a company like this in the market. We did not die. To the contrary, as we come here today, we are about a $700 million company. Our share price is about $12, and that's after the biotech meltdown of the last few weeks. And we have over $200 million of cash on hand. So we have everything that we need to really churn out innovation, to really build on what we've got right now and to broaden out those platforms. So is it too late? I think the answer is no. We had a great 2013. We were one of the best performing stocks on NASDAQ. Our ticker symbol is ARWR. But our innovation and our progress is now just starting. As you'll see, I think, in the next coming slides, we have an awful lot of value creation ahead of us, in particular this year, that we think will keep the stock moving. OK, so let's talk about what got us to this point and how we take this from $700 million to $7 billion. We are a targeted RNAi therapeutics company. What we did was we acquired Roche's RNAi business back in 2011. They spent upwards of $800 million to develop this program. So why should you care about that? What is RNAi? Well, it is a way of turning off target genes. So we know that many diseases are associated with genes that produce too much of some protein that is harmful. And we can turn those genes off. It is an extraordinarily powerful modality. So it is highly specific. We know that we can just turn off a single gene at a time if we want. It's highly powerful because no disease is undruggable. Many diseases right now are called undruggable because you can't get drugs to interact with the proteins of choice. That does not happen with ours. We can knock anything down that we want. Because it's selective, because it is specific, we can also limit off-target effects. And as you know, one of the big problems with drugs these days are side effects, and we can limit those with this modality. It also presents us with a faster development timeline. When you're talking about developing traditional small molecule drugs, it takes an awful long time because you don't know how you're going to perturb, you don't know how you're going to influence various cellular pathways. And the only way to know that is test it in animals and then in humans. In RNAi, when you're turning off genes, you know that if you design the molecule correctly, if you are knocking down the gene that is your intention, that it will work pretty much any time. And so once you have a delivery system, once you have a way to deliver these little molecules to the right cell at the right time, once that's validated, you can assume that that's going to work for any gene target you want. And so it gives you this amazing leverage that once you prove it once, you can do it 1,000 times with 1,000 different drugs. So what are we working on? Well, we have a pipeline, the top of which is against hepatitis B. We call that drug ARC-520. We'll go into that opportunity in a bit. We are in a phase 2 trial right now. Underneath hepatitis B, we have several other targets we have not yet disclosed. We're going to have a big analyst day in June in New York where we will disclose what the next drug is, but we have not talked about it yet. We've got 3 or 4 or 5 underneath this. We will have another drug that we will bring into the clinic by the end of this year and probably 2 more next year. And so we'll grow from a 1-drug company right now into a 4 or 5 drug company in the course of just 1 year. We think that hepatitis B is an outstanding first target for a lot of reasons. First, it is a massive market opportunity. Again, we'll talk about this in a bit, but there's about 400 million people on the planet that have hepatitis B, and there is no cure. Second, we have a potential for that cure, and we should know that in fairly short order because of the way our trials are designed. We should have a readout fairly early. And then finally, as I mentioned earlier, once we validate this for hepatitis B, you can assume that our delivery program and our technology is validated for other liver indications. Okay, so the investment thesis is as follows. We see a lot of near-term value driven by the hepatitis B program, and then longer-term value driven by the platforms. And those platforms are these, is our ability to turn off these genes. We have an awful lot of news events this year to continue to drive the stock. We're going to have this analyst day, as I mentioned, this quarter in June. We'll also finish our phase 2A trial in hepatitis B this quarter. We will read out data next quarter. We'll start our phase 2B multi-dose study in hepatitis B in the 4th quarter, and we'll have our next drug candidate filed with the US FDA also in the 4th quarter. All right, so why should we care about hepatitis B? It is a massive market opportunity. As I mentioned, 350 or 400 million people on the planet have hepatitis B. That's 1 in 20 people have hepatitis B. There is no cure. The current therapies require lifelong therapy, and those only make somebody less contagious, but it does not cure the disease. And so they still have the chance of getting cirrhosis, and they still have the chance of getting hepatocellular carcinoma. In fact, hepatitis B is the greatest cause of primary liver cancer. And again, there is no cure. This is 100 times more infectious than HIV. And so we saw this as a wide open opportunity. On the market side, we see hepatitis B as the next big value driver following after hepatitis C. So hepatitis C was a very large value driver over the last few years. Companies got into it and throw a lot of money into it. And now, at the end of that, we have a cure. As Jim Mellon mentioned earlier today, Gilead has a drug called Sovaldi that has been on the market now for for a quarter, and it sold about $2 billion worth of drug in the 1st quarter. I've never seen a rollout like that. It is just amazing. Now, the difference between hepatitis C and hepatitis B is that first, hepatitis C has multiple drugs that can lead to this cure. Hepatitis B has none, and so if we are right and we can get to that cure, we are the only game in town. Second, there are twice as many people on the planet with hepatitis B than hepatitis C, and so we see that as even a potentially larger opportunity than hepatitis C. So let's talk briefly about the biology of hepatitis B. Because I think it's important with respect to how we look to eradicate this. Once the virus gets into a liver cell, it does 2 things. It produces new virus like any other virus, and that makes you contagious. But it also makes an awful lot of certain viral proteins. And the importance of that is that those viral proteins suppress your immune system. So it keeps your immune system from clearing the virus. So that's the reason that we have some therapies that will be lifelong therapies that will keep you from being contagious, but will not cure it, because nothing we have, there are no drugs in development or on the market that can bring down those viral proteins. And so nothing will enable your immune system to come back up and clear the virus. And so that's what we're going after. So because we are RNAi, because we are able to turn off genes, we can turn off the entire hepatitis B genome. So what we do is we block both of these pathways. First, we block the production of new virus. And second, and potentially more importantly, we block the production of these proteins. And the theory goes, if you do that, your immune system will just naturally come back up and clear the virus and enable this functional cure. That's what our goal is. And if we can do that, we will truly transform this disease. And frankly, it will transform world health because of this massive problem. So can we do it? I think we can. We have an awful lot of data in animal models shown here, and I won't go through all the graphs, but shown here are some studies in rodent models. The takeaway message here is that what we were able to do is turn off the production of those viral proteins by 99.99%. We just silenced it. We just turned them off. We're able to turn off the production of new virus by 99.9%. We just turned it off. We did exactly what this promises to do. We just flipped the switch and it turned off. So that's interesting. No one has ever been able to do that in the history of hepatitis B research. This is the holy grail of hepatitis B research, and we're the only ones who've been able to do that. But it's a rodent model. So, is it translatable into humans? We think it is, but we don't know for sure. We then found a chimp with hepatitis B. So chimps are very interesting models because, of course, physiologically, they're quite similar to us. And they also get hepatitis B just like we do. In fact, they were used for the production of the hepatitis B vaccines, and so we know they're predictive of how of treatments for humans. So we found this chimp. It's got hepatitis B, and we were excited, and we wanted to test our drug on this chimp. That's the good news. The bad news was, it turns out this chimp had a mutant form of the virus that was a mismatch for part of our drug. And so half of our drug was potentially not going to work against this animal. That's okay, we were still interested. The second piece of bad news was that this chimp had a raging case of hepatitis B. She was far more viremic. She had much higher levels of virus than we ever expect to see in humans, and she had much higher levels of these viral proteins than we ever expect to see in humans. And so she was going to be a very difficult model, a very difficult patient to treat. I think of it as almost the worst case scenario. Even so, we decided to give it a try. Now, we were not going for curative intent, keep in mind. In the United States, it's harder to do chimp studies than human studies because of animal rights issues. And so we only had her for the course of probably a month and a half. And so we only could give her a small amount of the drug. What we think is that we need to give the drug probably once a month for 3, maybe 6 months to get to a cure. And we didn't have access to her for that long. But that's okay. We figured we can learn something from her anyway. And so we gave her the drug. And what we saw was shocking. And again, I won't go too much into the data unless you have questions at the end. But here's what we found. We found that we reduced her viral levels by about 95%. No one had ever shown that with any drug that quickly ever before. We showed the reduction of the key viral proteins that again, remember, immunosuppress the host and keep that immune system from clearing the virus. We decreased that by about 90%. No one had ever shown that before. So that was exciting. If that was all that we did, that was going to be exciting, and that was potentially world-changing. But we also saw evidence that we woke up her immune system. So shown here in shaded, the ALTs here. Again, I don't want to go into the science of it, but ALTs it's a liver enzyme, and this spike in ALT suggests that we woke up her immune system. We did exactly what we think you need to do to get to a functional cure. And so that was thrilling to us. No one had ever shown that before. It was tantalizing, though, right? Because it suggested that we were doing what we need to do, and it suggested that we entered this cascade of events that can lead to a cure, but we were not able to give her any more drug. Not because it was unsafe. We didn't see any signs of toxicity, but because of the crazy animal rights people. So we've done everything we can in animal models, and we've shown more than any other company in the history of mankind in terms of curing hepatitis B in animal models. And now it's time to go into humans to see if this thing actually works in humans. We completed a phase 1 trial in October of last year. That was in healthy volunteers. And a phase 1 trial is just designed to show safety. Does this hurt individuals? And what we showed was that it was extremely well tolerated. We saw no signs of toxicity. We saw no differences in, they call them adverse events or AEs. We saw no difference in AEs in the placebo groups compared to the drug groups. We just didn't see anything. And so it was as clean as we could have expected. So that was great news. And now the value driver there is that not only does that de-risk the drug, because now we're fairly confident that it's not going to do any harm, but it also de-risks the broader platform. Because remember, we are going to use the same basic platform for disease X, Y, and Z after hepatitis B. And if we show that this is safe in hepatitis B, it's also going to be safe in other drugs. And so we validated it across the board for safety. So that's good news. We then started a phase 2A trial in Hong Kong, and these are inpatients and actually infected hepatitis B patients. We started that the end of March of this year. And we have finished part of that trial, and that will complete this quarter. In fact, probably complete over the next several weeks, and then those data will read out in the summertime, maybe late summertime. So we're excited about that, because that will show, if it works like it worked in the chimp and like it worked in other animal models, that will show this deep knockdown, this deep reduction of this key viral protein that leads to a functional cure. And that will be a validator, not just for the drug, because that will de-risk the drug. It shows that it is effective insofar as knocking down these gene products, but also it will de-risk the platform again. Because if we can silence this gene, we can silence any gene. And so we believe the market will give us credit for our drug number 2, number 3, number 4, number 5 earlier than they would otherwise, because we have data to suggest they're going to work. And then finally, in the fourth quarter, we'll start a phase 2B trial, which is a multi-dose study, and it is in that study where we expect to start to get cures sometime next year. So what's the drug worth? We think a lot. If you look at where the drug is, where the disease is, the U.S. is actually a very small part of the overall world market. There's about 2 million people with hepatitis B in the United States. And so if you assume that you could price, say, a 50% premium to interferon, which is one of the standards of care right now, that equals about a $30,000 per course of treatment number. So that's a $60 billion market opportunity. And that's assuming, I think, very conservative pricing. As Jim mentioned earlier today, Gilead is pricing Sovaldi, their new hepatitis C drug, at $84,000 per treatment. And so I think we could probably price closer to there. But even assuming conservatively $30,000 for a cure, we're talking about a $60 billion opportunity. Then if you go to Western Europe, there are 14 million individuals in Western Europe with chronic hepatitis B. And if you ascribe the same metrics, now of course Europe reimburses for interferon less than does the U.S. And so if you assume that and assume a 50% premium, it's a $294 billion market opportunity in Western Europe. If you take that same measure and take it to Asia Pacific, ex-China, it's another 200 and change billion dollar opportunity. And then you go to China and discount it even more, we're talking almost $1 trillion market opportunity there. This potentially is the greatest pharmaceutical market that we have seen if we can get to these cures. So I think of us, and hold on to your wallet when somebody from biotech says this, but I view us as a value play. And here's what I mean by that. People look at us either as a product play or a platform play. If you look at us as a product play, if we're just one product, if we're only hepatitis B, given a $700 million market cap, I would argue that that is a discount to where we should be, given the size of the opportunity, given the dynamics of that market, given the fact that we have clear first mover advantage. There is nobody else in the clinic right now that has the possibility of these consistent cures like we do. I would argue that at $700 million, that is a value just based on that single product. If you then take that away and look at this as a platform play, just as a platform, you take away hepatitis B, look at what we can do to silence genes, cross gene, cross cell type. We also believe that it's a value. You look at our major competitor, a company called Alnylam. They're about a $5 billion company, and we can do anything they can do in terms of gene silencing. Potentially we can do it better because we have better delivery. We've shown much longer and much deeper reductions in gene silencing. But even let's assume we can only do just as well as them. They're a $5 billion company. We're a $700 million company. I think there's value there as well. So what's going to push us? We are a data-driven stock going forward. We have a great story, but we need to show that we are doing what we say we can do. And we've got great data coming out this year. As I mentioned, in the first quarter, we started the phase 2A trial in Hong Kong. In the second quarter, we will complete dosing that trial. We will also disclose our next drug candidate in New York this quarter. Third quarter, we'll have data on the phase 2A trial. And then fourth quarter, we will start the multi-dose study where we expect to start to see functional cures for hepatitis B, and we will also file an IND, which is a filing that you file at the FDA to push a new drug into the clinic. We'll also do that in the fourth quarter of this year. And then next year we have nothing but more data. We should have data on functional cures for the hepatitis B program. We should have clinical proof of concept for our next drug. We should also have clinical proof of concept for at least one additional drug. So again, while 2013 was a very big year for us, 2014 and potentially 2015 are even more full of data points that should continue to drive the stock. And I have no idea how long I've been talking, and I have no idea if I'm over or under time, but that's what I've got. And if we have more time, I'm happy to answer some questions. Do we have time? Yeah. Well, you ask a biotech CEO what's the worst thing that can go wrong, that's easy. We could kill somebody. I think that's not going to happen. So realistically, let's talk about our risks. So risk number one is the FDA makes you do all sorts of safety testing in multiple species to see if this drug is going to harm a human. But you never know if there's going to be some safety signal that is not predicted in an animal model. And so risk number one is that we see toxicities in humans that we didn't expect. I think we've gotten over that risk because the phase 1 trial was successful. We didn't see any toxicity at all, and the phase 2A so far, we haven't seen any toxicity either. So I think that risk is muted. Risk number two is that we don't silence the genes that we want to. I think that risk also is muted because other companies and we have shown that there is good predictability of data within this gene silencing from non-human primates and humans. So if it works in a monkey, if it decreases the gene in a monkey, it should decrease the gene product in a human as well. And we've shown that now across multiple primate species, and of course across maybe thousands of rodents, and we see it consistently. So I think the chances of us not knocking down the virus are very low. Risk number 3 is theory risk. So the world who thinks about hepatitis B thinks that if you can decrease these viral proteins, the immune system will naturally come up and clear the virus and get you to a cure. There's an awful lot of data to suggest that, but no one has ever been able to test it directly because no one's been able to knock down those proteins. So there is risk there, and we just don't know until we know. I view that as our biggest risk. I think that's also muted given the data around it, but that, of course, is a risk. Yep.
You're saying you just completely shut down that gene. Now, what about if the protein is just overproducing and you only need to cut half it? Can you do that?
Yeah, yes, we can do that. We can titrate this. It's harder for us to do because everyone's a little bit different. It's easier for us just to turn it off. But for those diseases that require you to knock it down a little bit, we can do that. I think that we will not get to those diseases for many years because there's plenty of diseases that we can focus on where if you can turn that gene off, then you're better. It's a much easier way to do it. It's much easier. And I'm a simple man. There's enough risk in this business that I look for our easiest route, and that's our easiest route right now. Yes.
Forgive me if I appear to be raining on your parade a bit, but I've been very interested in this area for 5 years, invested in it, investigated it extensively before I started, and RNA inhibitors is a cutting-edge part of contemporary science. There's about 60 companies worldwide in your space, the last time I checked, all working really hard on this area, and the main problem, as I understand it, is specific targeting. That's just to get the science done to where it's effective, and then you've got to go through your trials. You might have an advantage being an American with the FDA, but when I look at what AstraZeneca and GlaxoSmithKline have gone through, we've been told to go back and do new testing, and it's taken them $200 million and 4 years, not 400, God forbid, 4 years to finally satisfy the FDA that their drug is ready for approval. So can you just expand, because listening to that presentation sounds like it's a snap. You can turn off any gene anytime you want. You're doing it, and you're going to make billions of dollars just in the next couple of years. So you're a massively exciting investment opportunity. But could you talk more about the fences you are going to have to jump before you can actually bring this drug to market?
Yes. So you're right. The key challenge here is delivery. We know that if you can, so the molecule that initiates this gene silencing process are called siRNAs, or short interfering RNAs. And we know, the world has known for some time, if you can get these siRNAs into the right cell at the right time, you're going to silence that gene. It's a very robust, it's a very reliable system. The challenge is getting that into the right cell at the right time. And that's where we shine. Nobody, no company, none of those 60, none of them have ever shown the depth of reduction that we've shown. 99.99% knockdown. The world used to be excited about 60% reduction. Then they got to be excited about 80% reduction. And now sometimes some people can get up to 90% reduction. But I would caution that 90% reduction that some people get require very frequent dosing. Alnylam is a great example. They're able to get up to 90%, but they're talking about dosing once every 3 days. We can get 90% plus, 99.9% with a single injection that lasts 4 to 6 to 8 weeks. We also have some data showing that we can get it for 3 months in certain formulations. And so we are clearly ahead of the game in delivery, at least as far as published material has gone. And that's the great thing about biotech is that things get published, and so it's infrequent that somebody in their garage comes up with a new drug or a new technology that can scoop science. So I feel like we have a fairly clear path to get these genes knocked down. Now, that's not to say that we know it's going to be safe in every human. We don't know that until we know it. We have good data in the phase 1 and now in the first part of the phase 2 suggesting that it is safe. but we'll see if that continues. Regarding the FDA, we have had continued discussions with them as well as with other regulatory agencies, and we think that we have a good handle on what's going to be required. Europe, Asia, or various jurisdictions in Asia, Europe broadly, as well as the US. You mentioned GSK and AZ, so those are not RNAi drugs. They don't have RNAi drugs, and so those are a little bit different situations. But clearly, regulatory risk is always a risk in drug development. But we think that we have a handle on that to the extent that you can ever have a handle on government regulatory agencies. Okay.
One more question? Yeah.
Do you expect to have to come to the market again for funding in 2016 is the first question. And the second question, are you intending to bring the product to market yourself or with a manufacturing partner?
That's a great question. The first question is no, I do not expect to need money in 2016. We've got about $200 million of cash. That takes us through 2018 with our foot on the gas, just cranking out as quickly as we can, to mix my metaphors. So no, I don't expect any offerings anytime soon, at least that we need given our current burn rate. I'm sorry, what was the second question there? Oh yeah, partnering, right? Sorry. So that's a great question. To use Jim Mellon's story again, Pharmaset was acquired by Gilead a couple of years ago for $11 billion for the hepatitis C drug. And that wasn't an approved drug. It wasn't a phase 3 drug. It was a phase 2 drug. It was an early drug. But Gilead, which is a very smart company, saw the value of that and bought them for $11 billion cash. Well, Gilead traded down on that news because the world thought it was a crazy thing to do. It's too much money. Turns out they were very smart because they have created $80 billion or thereabouts of value based only on that drug, and they sold $2 billion worth of drug in their first quarter on the market. So that tells us that we should be in no hurry to partner this drug. I am perfectly happy to bring this all the way through registration and sell this drug. Now, I will not build a sales force in China. So we will certainly partner this, on a geographical basis. But could we build a sales force and at least co-market this in the U.S. and Western Europe? You're darn right you can, and I think there's a lot of value there. Okay, thank you. Thanks.